Hello students, today let us learn about embryo culture and plant organ culture. Let us start with embryo culture. Embryo culture is the use of plant embryo as explant and culturing them in vitro under aseptic and controlled physical conditions to obtain complete plantlets. The embryo at different developmental stages can be isolated and used as explants in embryo culture. The term embryo rescue refers to the culture of an immature or weak embryo which fails to germinate naturally to grow into a viable plant. Depending on the organ culture, embryo rescue may be referred to as embryo, ovule or ovary culture. Several factors affect the success of embryo culture, and the most critical factors are composition of the medium, pH of the medium, incubation conditions, time of culture, presence of suspensor in embryo culture, and the stage of embryo development at the time of culture initiation. Let us see the applications of embryo culture. The first application of embryo culture is in rescuing embryos from incompatible growths. One of the primary uses of embryo rescue has been in overcoming incompatibility barrier in interspecific and intergeneric hybrids where normal seed development often fails. The second application is in overcoming dormancy and shortening of breeding cycle. Embryo culture techniques can be used to shorten the breeding cycle in horticultural and crop plants with long periods of dormancy in seeds which delay breeding works. The third application is in overcoming seed sterility. The seeds of early ripening fruit cultivars are usually sterile because their embryos are still immature when the fruit ripens, and it is possible to raise seedlings from such seeds by using embryo culture method. The fourth application is in the production of monoploids. Embryo culture has been used in the production of monoploids from crosses between two species where chromosomes of one parent are eliminated in the process. The next application is in clonal micropropagation. Embryos have a high potential for regeneration and hence may be used as explants for in vitro clonal propagation. Let us now learn about plant organ culture. Different parts or segments of plant tissue may be used as explants and cultured on culture media to initiate cultures in plant tissue culture, and plant organ culture may be of the following types based on the organ used as explants. The first type is the shoot and meristem culture, second is the root culture, thirdly leaf culture, fourthly anther and pollen culture, fifth is the ovule culture, then flower culture and endosperm culture. The first type is the shoot tip and meristem culture. Shoot tip culture is the culture of terminal, that is about 0.1 to 1 millimeter portion of a shoot comprising the meristem, that is which forms about 0.05 to 0.1 together with leaf primordia and adjacent stem tissues, while meristem cultures is the in vitro culture of generally a smaller, less than about 0.1 mm in length structure from the shoot apex containing only 1 or 2 pairs of youngest leaf primordia. The main applications of shoot tip or meristem cultures are: first application is the production of disease-free plants. Apical meristems are generally free from viruses since viruses move readily in the plant body through the vascular system, which is absent in meristems, and a high metabolic activity along with a high endogenous oxygen level in the actively dividing meristematic regions prevent virus replication and multiplication. Therefore, culture of apical meristems is an important technique for producing plants free from pathogens including viruses, viroids, mycoplasmas, bacteria, and fungi. The second application is in micropropagation. Shoot tip or meristem culture of many plant species can successfully be used for micropropagation since they have high regeneration capacity. The third application is in the storage of genetic resources. Meristem from plants that produce highly heterozygous recalcitrant seeds can be stored in vitro at -196 degree for the long-term preservation of germplasm. The fourth application is their use in plant breeding. Shoot tip or meristem culture can be used to speed up breeding of hybrids in many plant breeding experiments where the hybrid plants produce abortive seeds or non-viable seeds. The fifth application is in quarantine. Since the plantlets derived from shoot tip or meristem cultures are disease-free and easily accepted by the Quarantine Authority for International Exchange, crop plants can be easily exchanged in crop improvement programs using this technique. The second type of plant organ culture is the root culture. Root culture can be defined as the in vitro culture of roots in a liquid medium under controlled conditions. Root cultures are generally initiated from the excised radical tips of aseptically germinated seeds, and the root tips are induced to grow like that of an intact plant by supplying the nutrients and hormones required for their growth. The root cultures can be multiplied by repeatedly cutting and transferring of the main root tips or of lateral tips into fresh medium in every subculture at the interval of definite period, and the culture derived from a single root culture constitutes a clone. Growth of the root cultures can be measured in terms of fresh and dry weight, increase in length of the main axis, number of emergent laterals, and total length of laterals per culture. Root cultures are important in providing information regarding the dependence of roots on shoots for growth hormones and knowledge of carbohydrate metabolism or role of mineral, ions, vitamins, etc. The specific applications of root culture are: firstly, root cultures can be used to study nodulation of leguminous root in culture. Secondly, plant can be obtained by regeneration of shoots from the cut ends of the cultured roots. Finally, another main application of root culture is the synthesis of pharmaceutically important secondary metabolites by the root cultures. The third type of plant organ culture is the leaf culture. Leaf culture is the in vitro culture of excised young immature leaf on suitable culture medium under controlled conditions. The mode of development of the leaf primordia in culture depends upon the size and age of the cultured leaf. Very young leaf primordia usually have higher regeneration potential than mature leaves. Importance of leaf culture. The first importance is culture of excised leaf primordia can be used to study the nutritional and environmental conditions required for leaf development. Secondly, leaf culture can be used for clonal micropropagation of plants by inducing the formation of shoot buds from the cultured leaf primordia. The third importance is leaf primordia cultures are also used to study the initiation and development of sporangia in fern leaves. The fourth type of plant organ culture that we are going to tackle is the anther and pollen culture. Anther culture is the technique of culturing excised anthers from unopened flower bud aseptically on nutrient medium, while pollen or microspore culture is the culture of isolated pollen grains. When cultured under suitable conditions, the microspores develop within the cultured anther or in the medium into callus tissue or embryoids that gives rise to haploid plantlets either through organogenesis or embryogenesis, and the phenomenon is known as androgenesis. Androgenesis may be of two types. Direct androgenesis, where the microspore behaves like a zygote and forms embryoids which gives rise to plantlet, or indirect androgenesis, where instead of undergoing embryogenesis directly, the microspore divide repeatedly to form callus tissue which differentiate into haploid plantlets. Generally, pollen cultures are considered more advantageous over anther culture, since overcrowding of pollen grains in anther and the growth of unwanted diploid cells from anther walls and other associated tissues is eliminated in pollen culture. Let us now see the importance of anther and pollen culture. The first importance is it is an important plant breeding technique to obtain homozygous pure lines, since anther and pollen cultures can be used for production of haploid plants in vitro, and homozygous pure breeding diploids can be obtained from the haploids by inducing chromosome doubling of the haploids by using methods like Colchicine treatment. The next importance is haploids derived from anther and pollen culture are useful in cytogenetic studies, and recessive phenotypic characters can be identified by comparing heterozygous diploid with haploid or homozygous diploid population. Thirdly, pollen cultures are suitable for studying the stages of androgenesis or factors governing androgenesis and other mutagenic studies. The fifth type of plant organ culture is the ovule culture. Ovule culture is the culture of aseptically isolated ovules from ovary on chemically defined nutrient medium under controlled conditions in vitro. Some of the main applications of in vitro ovule cultures are test tube pollination and fertilization. Ovule culture can be used to induce in vitro fertilization by germinating pollen in the same culture to induce pollination and fertilization, leading to the formation of mature seeds containing viable embryos. The next application is application in hybridization. Ovule culture has also been employed to obtain viable hybrid seedlings in interspecific and intergeneric process where the hybrid embryo fails to develop naturally. The third importance is in production of haploids. Haploid gallus can be produced by culturing unfertilized ovules, and these cells may be induced to form haploid plants by manipulating the culture medium. The fourth application is in the induction of polyembryony. The nucellus of monoembryonic ovules of plants like citrus can be induced to form adventip embryos in ovule culture, and the technique holds great potential in horticultural practices. The fifth application is in the study of zygote and embryo development. In vitro ovule culture also helps to understand the factors that regulate the development of zygote to a mature embryo. The sixth type of plant organ culture is flower culture. Flower culture can be defined as the aseptic culture of excised young or mature floral bud on suitable culture medium where they develop normally to bloom in the culture vessels and produce healthy mature seeds. Flowers can be cultured at different stages of development such as primordial stage, bud stage, pre-pollination stage, and post-pollination stage. Young floral bud and flower primordia requires a complex medium containing inorganic salts, B vitamins, amino acids, coconut milk, and growth regulators like auxins and cytokinins, etc. While the culture of mature flowers at pre or post-pollination stages generally requires comparatively simple media containing inorganic salts, sucrose, and low dose of growth regulators. The main applications of floral primordia or flower bud culture are: flower culture can be used for fundamental studies of flower development. The second application is culture of flowers before pollination may be used to induce the development of parthenocarpic fruits in the presence of auxins. Thirdly, the culture of pollinated flowers is important to study the process of fruit development and the role of growth hormones such as auxins, gibberellins, and cytokinins in the process. Fourthly, flower culture has also been used to study the sex expression in flower and floral morphogenesis. For example, in cucumber, the potentially male flowers tend to form ovaries in presence of IAA in the culture medium, while the addition of gibberellic acid counteracts the effect of auxin. On the other hand, IAA or gibberellic acid or cytokinins showed no effect in culture of potentially female or bisexual flower buds. The next type of plant organ culture is the endosperm culture. Endosperm culture in plant tissue culture is used for the production of triploid plants, since the endosperm tissues are triploid in their chromosome constitution. To obtain triploid plants, endosperms along with embryos are excised from immature or mature seeds and cultured on a suitable medium, and the embryos are removed after initial growth, while the endosperm tissue is induced to form triploid callus, which can give rise to triploid plants by organogenesis or embryogenesis. Let us now see the conclusion. Plant tissue culture technique can be used to obtain viable plants from weak and immature embryos which fail to develop into mature plants under natural conditions. Embryo rescue procedures have been successfully used for many years for producing interspecific and intergeneric hybrids and progeny of other incompatible crosses by culturing embryos or ovule and ovary in case of small seeded species. Understanding the nutritional requirements of young embryos along with modification of existing embryo rescue techniques should lead to successful application of these valuable in vitro procedures to important plant species. Tissue culture technique can also be used for culturing parts of an organ or whole organ of plants, and such organ cultures are useful for studying the developmental processes of the organs, since these cultures accurately model functions of an organ in various states and conditions by the use of the actual in vitro organ itself. Different organ cultures also find useful applications in plant biotechnology and breeding experiments like micropropagation, in vitro fertilization, embryo rescue, production of haploids and homozygous lines, etc. Thank you.